Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, we have a few announcements to make. The program of the third edition of the Dr. GPCR Summit is taking shape. Our confirmed speakers include Drs. Andrew Tobin, Ryan Roth, Rosie DeWalibi, Karen Gregory, Oliver Hartley, Ines Liebscher, Christelle Manet, and many more. This year, the meeting will be held between October 10th and the 16th. The meeting will be virtual to allow you to join us from anywhere in the world. Live talks will be hosted on Zoom from October 10th to the 15th, and everyone is welcome to participate by presenting a poster or submitting a pre-recorded talk. Similarly to last year, we will have presentation prizes for trainees. We're also trying out a few new things as well. We'll have networking and poster sessions on Wonder. We'll also have a full day dedicated to trainees. If you're a trainee, hurry up and submit your abstract today to give a talk or potentially present a poster. Visit drgpcr.com/summit today. We will also be hosting 3 workshops. We're excited to share that we'll have Dr. Sam Hoare and Dr. Luciana Leo, who will run a workshop on data analysis. Dr. Nicholas Smith will host a workshop on how to preserve data integrity in the lab. And this year, we welcome back the GPCR DB team with Dr. David Gloriam, Dr. Albert Koistra, Gaspar Nandi Sekeresh, and Jimmy Caroli. Everyone is welcome to attend the summit. And it's free when you become a Dr. GPCR Ecosystem site member, which is also free. Speaking of the ecosystem, we are excited to share that the Dr. GPCR Ecosystem 2.0 platform is now open. Visit drgpcr.com to explore the ecosystem today. Please note that we are slowly migrating from our old website to our new website. You'll always be able to find us at drgpcr.com, and we thank you for your patience during this transition period. The ecosystem, if you're wondering, is your GPCR-focused virtual playground. Join over 75 of your peers who have already registered as site members. Remember, it's free. You'll also have the option to select a plan and get access to all things Dr. GPCR and much more. You get access to the new podcast episodes with video before they get released to the general public, our new group discussions and forums, our job board, our learning center, where you'll be able to take a course or even prepare and share a course with your colleagues. And last but not least, you'll be able to discover GPCR companies and much more. Take advantage of everything that the new GPCR dedicated online playground has to offer today by visiting drgpcr.com and becoming a site member. Remember, it's free. Once you are a site member, you can also choose to sign up for an ambassador program, which means that you'll get your own dedicated Dr. GPCR ecosystem link. And every time someone subscribes using your link for a year, you'll get compensated. Last but not least, we're also looking for content creators. Subscribe to the ecosystem and start writing your own GPCR-focused content. Share it and show off your talents. You can always reach us at hello@drgpcr.com or visit drgpcr.com to find out more about all our activities. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR, and today I'm delighted to have with me Dr. Nariman Balenga. Hi, Nariman. How are you?
I'm good, Yamina. How are you?
I'm great. I'm so happy to have you on. I know we've been trying to get this podcast rolling for quite some time now, but we did it.
Yeah, I appreciate the opportunity. Yes, I've been waiting to talk to you for a long time. So that's amazing. I appreciate it.
Me too. Thanks so much for being here. So why don't we start with you giving us a short introduction as to who you are and what do you do?
So as you mentioned, my name is Nariman. I was born and raised in Iran. I did my bachelor and master's in University of Tehran. Then I moved to Austria to do a PhD in molecular medicine with focus on GPCRs. And then moved for a 3-year postdoc at NIAID in Bethesda, where I worked on GPCRs and the function of regulators of G protein signaling, RGS proteins in airways, especially in diseases like asthma. And then I moved to University of Maryland. I became an assistant professor at the Department of Surgery. Worked on adhesion GPCRs and one of them and what it does in parathyroid gland in the endocrine system. So over a year ago, I moved to sunny San Diego, started as a scientist in Ferring Pharmaceutical. Yep.
So I was listening and I have goosebumps because we have things in common. So let's
Oh, we do.
Yes, we do. Well, other than GPCRs, obviously. So you mentioned having spent time at NIH, at NIAID. I did spend time at NIH, at NIAID. So now I have to ask, whose lab were you in?
Or which building?
Yes, and which building? Yes.
Building 10, the medical center, actually. And then clinical center. So I work with Dr. Kirk Dewey.
Yes.
On the 11th floor.
Nice. I know who he is. So see, this is
we will get to it probably. Yeah.
I was at NIH not in building 10, but in the older side of the building. I was in John Curl's lab. And
whoa.
Yes, right?
Interesting. Yes.
Small world. And interestingly, I don't want to say something that's not correct, but I believe that Kirk Dewey is on a manuscript that we just submitted to Science with Helen Sue. And we're, I think, 86 authors on the paper.
Oh, wow.
And I'm co-second, fifth name. So see, it's very, very small world.
Yeah. Wow.
All right. What led you? Let's take a step further back. What led you from Iran to Austria?
Okay, that's a good question. So, well, I have to go a little bit back to high school, actually. So in Iran, so to get to college and start as a bachelor student, you need to take a test, and that's a national exam that maybe 1 or 2 million people, students actually take the exam, and they rank everything, and then based on the ranking, you can go to universities and different programs, or you can't go. So I got lucky back in 1999 because University of Tehran had initiated a direct PhD program in biotechnology, which meant that students with high ranking or students who had medals in international Olympiads could get to a direct PhD program. So they wouldn't need to take other exams from bachelor to master, or from master to PhD. So I took the chance. I went there. I joined a group of other 14 students, and we had, I would say, probably 50% biology, 50% physical chemistry, mathematics, heat and mass transfer, all those things that
we forgot about.
Yes. And so the point was to integrate biology with everything else, and to have students or future scientists who are innovative and collaborative, and they can collaborate with other disciplines. So that was the point of it. And then I did 6 years over there, which means probably a bachelor and a master's. But I had to take the actually a master thesis back then, which I did, and then afterwards, because of all the difficulties back in Iran and all the lack of resources and whatnot. So I decided to get out of that program, get my master degree, and go for a PhD somewhere else. So when I was doing my master's back at the University of Tehran, I had some interest in immunology and allergy specifically. And at the time, like 2003, 2004, DNA vaccines were a hot topic. There was no RNA vaccine back then. And so DNA vaccine is just, put the gene of a viral protein in a plasmid that then inject it to mice and see the reactions, type of immune response. So I was interested in allergy. Then I thought maybe we can put an allergen gene in the plasmid and inject it to mice and prevent allergy later on after sensitization. And not only that, but also I wanted to mix the plasmid with some type of nanoparticle or something like that prevents the sudden release of the plasmid in the tissue. So it would be a gradual release. so which will prolong the type of immune response that we want.
Yeah.
So I had to start everything from scratch, find the lab that I can work on this project, find the plasmid from somebody else abroad, all of that. And so I ended up working with mice with a DNA vaccine for an allergen from Birch pollen, actually, and mixed it with nanoparticles, injected, and looked at all the immune responses. So it was fun. It was very exciting at the time. But then as I said, I wanted to go for PhD somewhere else.
Yeah.
Applied many places, some in Europe. I actually got a couple of admissions from Germany, Switzerland, and Austria. And then that's actually where I ended up doing GPCRs, because I had never heard of GPCRs, to be honest with you. And then I had admission from different places, and I had to make a decision. Most of those programs were actually in the same field, like DNA vaccines and vaccines for parasites and whatnot, dendritic cells, all immunology based. But just an accident, literally an accident, led me to work on GPCRs. So the PI in Germany had an accident the day before I make a decision in a ski resort, and she completely got paralyzed. So I couldn't go there. I couldn't start that program. Yeah. So I had one option, and that was going to Dr. Maria Waltor in Medical University of Graz and work on GPCRs, which I'm really happy that I did. Not only because of working on GPCRs, but also working with Maria. She's a great scientist, a great person, actually. So that's how I ended up starting a PhD somewhere else.
In the field, and in the field. So, because you mentioned you never heard of GPCRs before.
No idea.
So let's before we move forward, let's take 2 steps back. So you mentioned really your interest in science, but as a child, were you interested in science, or did you know what you wanted to become?
Well, science, I don't know. I was curious, I would say. Luckily, we didn't have iPhone back then, so we had to do something with our cousins and friends and whatnot. Always outside. I was a good student. My grades were good, but we had plenty of time at our hand. So I don't know. We made a lot of things like a walkie-talkie and whatnot, and destroying radios and putting it back. All of that. That sense of curiosity stayed. Otherwise, I wasn't really thinking about science back then. But yeah.
Interesting. I like to ask these questions because I love the breadth of responses. Some say, well, I was interested in physics, or some say I was interested in, becoming an astronomer, or some say I have no idea what I wanted to do. I was just very good or not so good at school. And it really is dependent. I was thinking about it, and I was trying to figure out if all of us scientists who I talk to, do we have something in common? And I think curiosity is that aspect.
Yeah. Well, I mean, the last couple of years of high school, I would say genetics and that type of biology that was introduced to us. So that was quite interesting, right? So before that direct program was available, it would be just, going to middle college school. And I didn't want to do it, really. At the same time, I wanted to follow biology. So I had no option until I got lucky for that program.
That's amazing. And I'll see. And why immunology? You mentioned asthma and inflammation. Was there a particular reason, or you just find it interesting?
Yeah, a little bit of personal reasons, and also during the immunology courses that we had, I was fascinated by the type of cell type. I mean, all the cell types and how they all communicate together. From dendritic cells to T cells and neutrophils and eosinophils. It just amazed me, and I thought that system still is. Immunology is one of the most evolving parts of biology. Every day there's a new cell type with a new marker, which does something completely new. So that was the reason.
Yeah, I think it's a messy, although it's a beautiful field to me, it's just there's so many things that happen, and if you don't follow, you're done. The other day I
Yeah.
had to print out a paper, a review paper from 2015 that had all the CD markers because people, immunologists talk about all these CD things, and I'm like, okay, what is that? What are you talking about?
Yeah. Yeah. No, no, that was fun.
And so you end up in at the University of Graz in Austria, and you knew that the lab you were joining, so you knew that it was going to be about GPCRs. But what was your first impression of the field of GPCRs? Were you thinking, "Oof, that's a new, different world," or "I prefer immunology," or how did that, what was that cultural shock science wise?
Um, I would say that Maria was extremely helpful on that
She's
aspect.
amazing.
So she, I never forget. So she gave me 5 pages, each one with one project on it,
Hmm.
and they were all about GPCRs, right?
Obviously.
And um, and then I had to choose between them. So the first thing I did was just put those keywords in the PubMed and see how many papers out there. Four of them, hundreds of papers were coming out. And then the other one, the last one was GPR 55. I put it in there and there was just two papers. And then I Googled it, there were two patents, and that was it. So I thought to myself, okay, if I choose those, I'm going to read hundreds of papers. I'm not going to do that. So let's choose something that is, almost nobody knows anything about. So let me start with that. I knew it's going to be difficult, but I knew that at the same time I will learn the most probably from A to Z. And she completely approved that, and she supported me all the way through those 3.5 years. Um, so getting out of that, immunology and that kind of in
Yeah.
vivo studies and completely focusing on one protein and all the signaling pathways, it was new. I mean, yeah, I did not expect to be doing that at all. But again, the environment was so good that I think I accomplished a lot back then during that, yeah, 3-4 years. Um, and I really like GPR 55 because it helped me to understand GPCRs to
There
begin
were
with.
two papers.
Yeah. Um, yeah, I would say it was quite a fun experience. And so we had to, so based on the patents back then, it was supposed to be a third cannabinoid receptor.
Okay.
So we ordered a lot of endogenous cannabinoids and synthetic cannabinoids, and the, the assays that those patents, which are usually gibberish, uh, were providing were, I tried all of them. I spent probably 6 months chasing a white goose, and I got no data. And then I was saying, "Oh my God!" I think this has happened to many students, PhD students.
Yes.
Yeah.
Yes.
Until the day that we just switched some of the assays and we got the signal, which was quite a day. So we found that there are some inverse agonists of CB1 receptor, which are actually agonists of GPR55.
Interesting.
Um, and then during this time, while I was working, a Japanese group actually found that a lipid, um, lysophosphatidylinositol, is actually one of the endogenous ligands of GPR55. So having those agonists, it helped a lot to further the studies. I went beyond what was the initial goal of the project. We looked at heteromerization of GPR55 with CB2 receptor in neutrophils, with the CB1 receptor in endothelial cells. So that was just an amazing experience with Maria, and yeah.
It's so interesting when you mentioned that, you had all the five projects, and then you had the one that had the least papers. And in my mind, while you were speaking, I was like, okay, you're gonna tell me that you're gonna pick the toughest one because that's the most challenging. I never expected you to say that there's just two papers and it's less time to read them, knowing obviously that that's a challenging
Yeah.
project.
Yeah. Yeah. So back to your question, how I kind of managed to go out of immunology and
Yeah.
get to this. So at some point during this project, I asked Maria whether I can work on neutrophils because I had figured that neutrophils express GPR55, and that immunology background always pushed me toward this. And then she said, "Of course, you did all the other things. Go for it." So yeah, and I looked at the reaction on response of neutrophils to LPI or this endogenous ligand, and also a CB2 receptor agonist, and how they actually crosstalk, how they modulate the migration of neutrophils. So that was
How
fun.
interesting. Okay, so let's kind of talk a little bit more about so moving from Tehran to Austria. How hard was it to adapt? It's a different country, it's a different language, it's a different continent, it's a different culture. And I'm asking this one because I'm interested in your answer, but also I'm thinking about the listeners, especially the trainees who are thinking of going to do a postdoc or going to do a PhD in a different country. And I think it's interesting to hear other people's experiences and learn from them.
Yeah, so I think initially, maybe a couple of months, it was difficult to just adapt to the situation, but I had the best colleagues, the best mentor. I remember a lot of times I had to do some official things, and all of colleagues helped me in person, coming with me. So I didn't know the language, right? Yeah, so I think the environment that you work in is absolutely important. Your mentor, not only being good scientist, but also being considerate of your condition. That's absolutely essential. So all of that helped us. I mean, even I had my kid born in Austria. So you can imagine how it was at the hospital. You couldn't speak to anybody. But all in all, I think that was a great experience. Yeah.
I think it's always rewarding, and it may be hard in the beginning until you get used to things, but I think it builds character. Moving. I remember a podcast episode I recorded with Khaled Al Abdurrahman. He is a postdoc in Steve Ferguson's lab, and he moved from Egypt to Canada. I think he was in December to Manitoba. So you can imagine the shock in just the shock in temperature.
Yeah, I can understand.
But it's still, I think it's worth pushing yourself out there into that discomfort, because that discomfort is temporary.
And what helps a lot? Some sort of self-motivation. If you are motivated to reach a goal, or to discover something, and it will help you. It will help you during these moves. It will help you during the stressful times of funding and papers and defending your thesis and whatnot. You need that in addition to everything that the environment has to provide, encouragement and support.
Yeah. But I think as PhDs in general, we do have this ability to take on a lot of bad stuff, like experiments not working and cells dying and all of these things, because, and I think about myself, and we live from dopamine rush to dopamine rush, when something finally starts working, and then you have to, savor that moment, because you know that for the next 6 months, stuff is not going to work.
Yeah, that's true. That's true.
And so after your PhD with Maria in Austria, you moved to NIH. How did that move happen, and why did you pick NIH?
Well, I picked NIH because I would say it's probably the best place to work as a postdoc. Yeah. You know better. So actually, all the core facilities are amazing, excellent. The environment, the resources, almost unlimited funding. You don't need to write anything. You don't need to take any courses. You don't need to teach. All of that allows you to, just go full of speed. And Yeah, and I knew that because I had a few people who had been to NIH, and
Hmm.
so that was the main reason. Now, why I joined the lab of Dr. Kirk Dewey is because he was working on allergy and GPCRs. So back to the main topic. Yeah, so it was a dream come true, right? So, and he was working on mouse models of asthma, specifically allergic asthma, because we have different types of asthma. And I mean, he's one of the top scientists on RGS proteins,
Hmm.
regulators of
Yeah.
G protein signaling.
Yeah.
So it was the best place to go for me. And yeah.
And how was the transition? So, for the sake of trainees,
Mhm.
once you were finishing up your PhD, did you look at different labs, and what made you pick Kirk Dewey's lab?
I did look at different labs. So it was me and my wife, right? So we
Yes.
were applying at the same time because she's a scientist as well. She almost finished her PhD at the same time in the same university.
Mhm.
Um, I was very picky. After getting all the background on GPCRs and knowing my interests from past, I was quite picky of what lab I want to go. Um, I applied different places. You named John Carroll. I actually applied to his lab as well. Um,
See,
um,
small world.
yeah. Um, but yeah, it just worked out for me
Mhm.
in Dr. Dewey's lab, and also my wife was able to get the postdoc position somewhere else at NIH. So that was
That's
just
great.
yeah.
That's great. And I think thank you for sharing the fact that you and your wife are both scientists, and a lot of couples scientists have this problem whenever it's time to switch, is that you're not looking for one role, you're looking for two jobs.
Hmm.
And
Yep,
that sometimes can be challenging.
that's true. That's absolutely true.
So at NIH, going from Austria to the US, what were your observations? How easy was it to adapt, or did you have the same ability to, 6 months in and you were okay? Well, the
Um,
language was not much of a
that made everything easy, right? I think it was okay. Lifestyle was very different.
yes. It's Maryland.
um, yes, but I mean US versus Europe, right?
Yes, that's true.
Yeah, so the hours of work and things like that. But for us, it didn't matter. I mean, we were quite motivated to work as many hours as possible. I
Yeah.
didn't really care about that. But generally speaking, it was a little bit tense initially, but overall, I think we managed it. So we stayed there in Maryland for about 10 years, and we were at the same place, basically, NIH, a couple of
Mhm.
different buildings. Um, I think the transition was okay. I have to say, the work in Dr. Dewey's lab for me initially was quite challenging because I had to do a lot of in vivo work.
Mhm.
In addition, I had to develop a new ex vivo assay. So I finally managed it. So I was basically solidifying mouse lung and then slicing it and then inducing some contraction of the airways and
Wow.
imaging it. So it was quite difficult to get it right. So the first 6 months probably was very challenging, at least at the science
Yeah.
part of it. But overall, it was okay.
Yeah, I think it's a big change going from Europe to the US, or from Europe to North America, although there's differences between Canada and the US. But I think moving to Maryland was a huge challenge, because we lived in New York for 5 years, in Manhattan for that matter. And then you move to Maryland and it's quiet. People complain about the traffic in Bethesda. No, there's no traffic compared to New York City. What are you talking about?
Yeah, yeah,
But
no, I understand.
it's a completely different world. But I do get the change. And then so in the Drury lab, you worked on which GPCRs did you work on?
So it was not specifically any GPCR. So we were working on RGS4 and
Mhm.
RGS5.
Okay.
We had global knockout, tissue-specific knockout. Um, The lab was focusing mostly on airway smooth muscle cells that actually induce the contraction of the airways in asthmatic patients. Um, and we had these mice. I was developing mouse models of asthma by injecting, and then intranasally challenging the mice with different allergenic resources. And so we would look at the response of the cells to histamine, acetylcholine, thrombin, everything that is actually present in or elevated in the lung of an asthma patient. And then looking at how RGS4 and RGS5 can actually suppress the hyperactivity because they work as mitigators of GPCR signaling. So that's part of the things I did in Dr. Jui's lab. And, as I mentioned, the resources at NIH are unlimited and the facilities are great. You just give the cells to them. They know what to do with it, whether it's confocal or anything else. So I was feeling like this is not enough. I need to do more than this. And then at some point, I came up with the very naive idea of because we were developing these mouse models by intranasal challenges with some fungal allergenic source. So it was Aspergillus fumigatus, which is a mold that is everywhere. It's ubiquitous. So probably you and me, we are inhaling it right now. But most of us don't get asthma. And then I was wondering, maybe this source that we are challenging the mice with, maybe these allergens are able to actually reach the smooth muscle cell layer in the lung. And I told Dr. Jui, can I follow up on this? I just don't know what they do if they reach that layer in the lung. And he said, yeah, you are done with other things. Go ahead.
And then same like Maria, you're done.
Exactly. Yeah, so that's why I would say it matters who you work with. So if they provide the environment for you, you would just, flourish. So I took the chance. It took me 2 years to figure it out. But finally, we found that there is a protease in this Aspergillus extract. It's a serine protease. It's called alkaline protease 1 or ALK1 that actually degrades the extracellular matrix around the smooth muscle cells. By doing that, it prevents the integrin signaling, which through a very complicated mechanism unleashes phosphorylation of myosin light chain. And myosin light chain phosphorylation is a hallmark of contractility. So basically adding that to the mice, we were able to increase the contractility of the airways. So they were responding really highly compared to mice which are not, challenged by that specific protease, which was great. It was in vivo and everything, but it became really great when I saw that our antibody was detecting that specific allergen in the lung sections from asthma patients with severe asthma. So that was one of those aha moments that
I ask about.
Okay. It recapitulated everything that we were doing, so that was quite amazing.
I like these experiences where actually, you do an experiment and you know that you're the first one who has this answer, but then I think in order to like the topping on the cake or the cherry on top of the cake is really to see that what you see in animal models is actually translatable to patients.
Yep.
Because at the end of the day, that's why we're doing what we're doing.
Yeah. But it was a crazy idea at the beginning. I didn't know what I'm doing, but he just let me do it.
You had a hunch.
Yeah. No.
You had a hunch, and I think that's something that a lot of scientists can and should and are following. Sometimes it tells you, meh, your idea wasn't good, but sometimes you make these
It can happen. Yeah.
connections.
Yeah. I mean,
And
there would be failures, of
of course,
course, but yeah,
but
sometimes you get lucky.
I think this comes also with experience. Is when you've worked in in a field or on something for a long time. At some point, you get these hunches and you have these ideas and you formulate these hypotheses and you're like, okay, let's just try it out and see what happens. And I think having that ability, as you mentioned, to do this at NIH, where you don't have to.
Yeah, you don't worry about anything, right?
Exactly. And I think it's really nice to have a mentor that supports you as well, who says, yeah, sure, go ahead, let's try it out. And it's absolutely worth it. Excuse me. So after your time at NIH, I'm sorry, I need a sip of coffee.
Me too.
All right, there you go. And then you decided to move on and look for an academic position.
Yes.
What was the motivation behind that?
Well, okay, let me be honest. After finishing, well, I stayed at NIH for 3 years.
Mm-hmm.
And usually people don't do that. People who are visiting fellows or postdocs, they stay 5 years, they turn that to a 7-year, because why would you leave NIH? But for me as Iranian, I was really into getting my green card. And I could not do that while at NIH for many laws that prevent that.
Yeah, the type of visa I guess you were on.
Yes.
Went through that as well. We have that in common too.
Okay, see, there you go. So I had to leave. And I wanted to, because that was important for my family. So I found a lab, or actually a department in University of Maryland, who someone was working on RGS proteins, but in a completely different system, endocrine system. So I joined that lab and department, and very shortly after that, I became an assistant professor working on parathyroid glands, endocrine system, and yeah,
I think that's really an important point to raise for all trainees, especially if you come to the US. It's such a process to be able to stay here long term if if that's the plan, or at least to have the ability to do that. And I went through the same thing. I went for a postdoc in New York. I was on a J1. You can't, I didn't know what I wanted to do, full disclosure. And when my time was up in New York, I ended up moving to Maryland and working in John's lab. And it was this whole interesting situation. I went to a conference while in Tom Sackmar's lab, and I had a poster. Someone came up to my poster, said, "Oh, my roommate needs to talk to you. This is amazing." So I ended up talking with the roommate. His name is Ian, and Ian was a PhD student in Helen Sue's lab. We ended up collaborating. Helen introduced me to John, and that's how I ended up at NIH with a dual intent visa. And as a result of that collaboration, we just submitted 2 or 3 weeks ago that paper to Science. So it's all intertwined. In there. But that's the time at NIH was the type of visa that allowed me also to go, and I was able to do cool science as well.
Yeah. So if you're coming from somewhere else to US, you gotta be prepared for those things. And
yes, you have to know ahead of time what your intent is and what you want to do and how you want to prepare for it. I think it can be easier if you go to Canada, for example. I had a colleague who came for a postdoc. This was over 10 years ago, maybe 15 even. And 6 months after she arrived, she applied for her permanent resident card, got it done, which was, I want to say, much simpler than the US system. But this is just, for people who are listening to think about planning ahead, not only from a scientific perspective, but also from all the life perspectives.
Yeah, and those things, normally you wouldn't know unless you talk to somebody like you who has gone through it,
Yeah.
So the more you talk to people, the more experiences you
hear about.
Yeah.
Which is why we're doing what we're doing here.
Exactly. There you go.
So after the University of Maryland, you moved to sunny San Diego. What made you, if you don't mind me asking, what made you do the switch?
The switch, okay, so when I got the position at the University of Maryland, I was still thinking about going to pharma.
Hmm.
I got that position there, and I didn't reject that,
Of
declined that.
course.
So um, simply because type of background, type of skill set that I had obtained over the years was very welcome in pharma.
Hmm. Yeah,
Um, Whether it was therapeutic area or GPCRs and whatnot. Um, and beside that, I did not want to write grants for 30 years. It
yeah,
was not, I can easily say that it's a dysfunctional system. I've been in and out of it. I got my grants, my R1, my funding from other foundations and whatnot. I had plenty of budget to work
hmm,
with, but it was not something that I was enjoying the process.
yeah,
And I did not want to do that forever. And also, I knew the type of work that I'm doing. I can do pretty much the same thing in pharma. And at the end of the day, I know that this is much closer to the patient, and it will probably help if it doesn't fail. Um, so there was no reason why not go to pharma for me.
yeah. Yeah, I think you made many important and I think interesting points. One goes back to knowing what you like to do, and you very quickly identified that writing grants for the next 30 years is definitely not something that you enjoy doing. And also moving into industry, I think it's important for people to think about industry. For a long time, at least when I was growing up scientifically, it was, oh, the dark side of science, which is not true. I think it's the other side of the same coin. And yes, when you work in the private sector, the way people work is different because there's deliverables, there's other priorities there. But at the end of the day, the science, the soundness of the science is just as important as doing it in academia, even if not more important.
It is more actually. Probably, but we spend a lot of time just reproducing what's in the papers. Um,
Yes.
and
Yes.
um, and we have to prove everything, every single item. So I'm doing pretty much exactly the same thing I was doing back there,
Hmm.
either at the bench or meetings and whatnot. There are more meetings. You
Yes.
are not scheduling the meetings. You
Yes.
have to jump from one job to the other, multitasking. Yeah, that's there. But I think that, so. I mean, I've been here for a little bit more than a year. Um, I think it has been a great experience.
Great.
You get, yeah,
I think well, I spent way more time in academia than outside of academia, but definitely it's a different pace. Being in the private sector, priority, timelines are shorter, and then it's bang bang bang. But what I did like about industry is that budget wasn't a problem, you know and the whole idea was not to show that you can set up an assay, but you need the data.
yeah,
You have to make sure that the assay obviously is sound, it's reproducible, but that's not the most important thing. The important thing is beyond that is really the data, so that you can move forward from milestone to milestone. And I think
correct.
that's very
correct.
important too.
I agree.
All right, let's go back to GPCRs. What is your favorite one? My guess is going to be GPR 55.
You nailed
Or
it.
no.
Yeah, so to be specific, yeah, it's GPR 55 and another one that I worked at the University of Maryland.
Mhm,
But I would say generally, I would say orphan GPCRs
mhm.
are the most interesting one for me because I always think that these orphans are expressed in some specific cell types. Why are they there? Why a cell would, expend energy to make that protein to be just there? There is a reason for it. So that excites me. It's been 15 years now. That's the most interesting thing for me. And now we don't know their function, we don't know their signaling because we don't have a ligand for it. You have to find a way to, get a signal from it, monitor the signal, and look for the function, whether it's knockout and whatnot. So that in general, orphan GPCRs. But GPR 55 is definitely my baby, so I'll love that. The other orphan GPCR that I worked with was an adhesion GPCR, ADGRG2 or GPR 64, and I worked on that specifically because I was working on parathyroid glands. So as you know, parathyroid glands regulate the body calcium homeostasis by regulating how much parathyroid hormone is secreted. Now everybody for the last 20 years thinks that it's calcium sensing receptor that is detecting the amount of calcium, then regulating how much PTH is released. So we had some transcriptomics data that showed that there are other GPCRs, and one of them is almost exclusively expressed in parathyroid, and that was GPR 64. And again, I put it in the PubMed, and nothing came up.
So you pick the receptors for which there is barely anything.
I think they are following me. So and then, so that was like 2015 or something. But I was lucky because this is one of the adhesion GPCRs, which has 33 members. But I was lucky because a couple of years before, some folks in Germany and US actually figured out the overall or the general mechanism of activation of these receptors. They have N-terminal fragment that is detached from the rest.
And
exactly.
The whole
yeah, so I started to learn, and I just suddenly understood that I know nothing about this class. I had never heard of them, maybe. So I started looking into that. A lot of pharmacology without even having a ligand to make it to be active, to look for activation. As I mentioned, I was at the Department of Surgery, so I had this opportunity to get freshly resected parathyroid tumors or normal parathyroids from cadavers and take it to the lab, isolate the cells, miniaturize the assay in 384, and then look for the signal. So that was quite fun. And then, so that was about 6 years. So that's the other, favorite GPCR.
GPCR, yeah. I think adhesion GPCRs are very interesting class. And when I recorded my episode with Ines Liebscher and Simone Premel, I was like, wait a minute, oh, they do all of these things?
Yeah, and they are expressed, in so many weird places: tonsil, ear, parathyroid, epididymis, synovial joints, these places that we never knew that, there are GPCRs functioning there. So it's
a GPCR. Well, they're everywhere.
Everywhere.
Any disease, anything, or physiological response, there is a GPCR doing something there.
Doing something, yes.
But I think it's a really interesting class of proteins, and also a very interesting they not only their physiological function, but they're the tools that we need. There's many more tools that we need to develop to better study adhesion GPCRs. And I think that's the fun and challenging thing at the same time in the field.
Yeah, exactly. And I think one of the main questions for adhesion GPCRs is, so they have these N-terminal fragment, right? And we don't know in most cases in the diseased state in a patient if that N-terminal fragment is still there or not. Are we supposed to target that, or are we supposed to target other domains? So that, I think that's something that has to be figured out.
And also if that domain is cleaved off, does it go activate something else? Where? And
it's open field for people who work on adhesion GPCRs.
Yeah, I think so too. I think it's a very interesting field to be into, and I think I'm most fascinated by the size that these receptors can have, 600 to 6,000 amino acids. I mean, it's just, what do you do with the...
Yeah, the Western blots are also challenging, I can tell you that.
I can imagine. I mean, I had trouble. That's just it's one thing. I always was poorly skilled at doing Western blots, even with your typical class A chemokine receptor GPCR. I would always get different results every time. Same lysate, same everything. And at some point I said, you know what, I think I'm gonna stay in live cells and not do Western blots.
Yeah, maybe your antibody was not good. Let's say that.
Maybe it was a tag antibody, it was a 1D4 antibody. But I don't know.
Okay.
And it's funny because the purified rhodopsin control always looked great. Same thing. It was just my GPCR. So I don't know. But I can only imagine the challenge with, a 6,000 amino acid receptor there.
Yeah, it gets ubiquitinated, a lot of glycosylation and whatnot. So
Good luck even purifying it.
yeah.
Absolutely. All right. So we talked a little bit about some advice for junior scientists. The fact that you have to put yourself out there, travel, meet with people, talk to people, and then how to adapt to a new environment. Do you have any other advice for junior scientists who want to contribute to the field or contribute to science in general?
I have some probably suggestions.
Please.
You know, all of us come from academia, right? I mean, we should have done academia to come to pharma.
Yeah.
But not many people go from pharma back to academia. So for students, postdocs, my suggestion would be to talk to as many people as possible. Get the insight of people who are in pharma. And then make a decision if you want to do it or not. Also consider that, contributing to human health is not always through doing experiments in the lab. There are a lot of other options out there, whether it's regulatory efforts or whatnot. It's not always experiments. You can maybe contribute more. And sometimes you even don't know what talents you have because you have not tapped those potentials. So try different things. Don't limit yourself to just pipetting. That's not what it is. Yeah. So
I think I was just going to say, I think that's fantastic advice. And I think that's very important. And I wish somebody had told me this maybe 10, 15 years ago when I wasn't sure what to do. And in my mind, it was, I can only become a PI. And it was through more hours pipetting in the lab. And
yeah.
that's now looking back, it's not true. And you can be basically anything you want within the scientific field without a pipette as well. I like that you mentioned regulatory affairs, but you can become even a patent agent, a patent lawyer if you want to go to law school after that. I don't know, after a PhD who wants to go to law school, but hey, you never know. But there's other things that you could be doing to contribute to health.
Yeah. And, I think I mentioned that choose your mentors or your managers very carefully. And also the place that you are going. It's not just a lab. Sometimes the university, department, all of that comes into play.
Yeah.
How they encourage you, whether they allow you to, find your own niche, even if you are staying in academia. Yeah. So I know it's difficult. Many times it's very difficult, but the more you talk to people around, the more you understand about people. A good scientist is not just a good scientist. It needs to be he or she needs to be a good person. So otherwise, yeah.
Yeah, I agree. Perhaps if you can expand on contacting and talking to people from a tactical perspective. How would a PhD student or a postdoc go about that? Because we all talk about networking, and I hate this word because it feels so not I don't want to use the word sneaky, but it's like so pushed forward when actually networking it should be this natural progression of things. We are networking right now because after this I'll know things about you and you'll know things about me, and that forms a connection.
Yeah.
And I think
yeah, so I mean, generally speaking, I would say when you go to conferences, don't be shy. Go to, I mean, if you are interested, out of academia, go to posters that are provided by even pharma, or just find those people from FDA or other places
Yeah.
and just talk to them. What do you do per day? What's your routine? Um, so that's one thing. And also, I know that, I mean, at the University of Maryland, that was the case. I know many universities do that. The last few years of PhD, they invite people from outside. So be active in those meetings. Don't be shy. Ask questions. Um, you will get some sort of idea of what you will be doing in pharma or other places.
Yeah, agreed. Agreed. I think it's important, for example, at NIH, I remember there was a whole panel with different people from AstraZeneca. Actually, it was MedImmune at the time, but from AstraZeneca now.
Maybe we have been to the same meetings. I don't
I
know.
know. I know. I wonder. Actually, this is what I wanted to ask. When were you at NIH?
2011 to 2014.
Okay, so we missed each other by 2 years. I started at NIH
Oh,
in
okay.
2016. So
Okay.
maybe the people from MedImmune slash AstraZeneca were there multiple times.
Yeah, that's possible.
So yeah, but that's also important to just go out there and ask questions. And I say this every time we talk about this young advice. People are 99% nice. You just have to be genuine and say, look, this is my situation. I'm finishing up my PhD. I'm looking at my options. For example, on LinkedIn, your profile seems interesting. I was wondering if we can have, a 15-minute conversation and keep that conversation on that person because you want to learn from them.
Exactly.
And
Exactly.
you
Don't
never
don't
know.
take your resume necessarily. Yeah, just
No,
talk about no.
no, talk about that person. Do not say, I need a job.
Yeah, yeah,
Just get to take notes, get to talk to different people in a role that sounds interesting. And then you can, from there, learn a little bit more and then make your decision. And you never know. Maybe you're going to talk to someone, you click with them, and they're going to be like, oh, send me your resume. You don't know.
Yep,
But that shouldn't be the number one objective. The objective is to
exactly.
learn more about the person.
Well said. You said it much better than I would say.
Thank you. So we talked a little bit about your work on adhesion GPCRs and on orphan GPCRs. Any aha moments that you had as a scientist that shaped the way you see science or GPCRs in general?
Yeah, so I think I mentioned a couple of them.
Yes.
Yeah, after the first 6 months of PhD, when I found the signal from GPR55, that was quite a day. And I kept doing that assay forever.
What was it? What was the assay?
It was a luciferase-based reporter assay. So we were looking for a calcium response from this receptor because the patents were saying there is a calcium response. It's a GQ coupled GPCR. In our hand, it was not a GQ coupled GPCR, but it was inducing a calcium response very slow through G alpha 13 because we still think that GPR55 is uniquely coupled to G alpha 13. But it was through ROA, ROC, and phospholipase C epsilon, completely different pathway. But then we talked with Maria and we figured that maybe NFAT, nuclear factor of activated T cell, would be a good, indicator of that type of calcium response, and it was. So that, that was one of them. The
How
other
did
one
you
was,
celebrate before you move on? How did you celebrate?
well, you have been to Europe, right? So
Yes.
the cold rooms in Europe in the labs are full of
Alcohol.
booze. Yes, we don't keep reagents there; we keep booze there. So yes,
We
that
did that
was
in
the way.
Montreal too.
Oh yeah, I
Oh
didn't
yeah.
know.
We did that. We had, parties and things like that in the lab. Well, not in the lab, but in the group. And the beer, the champagne ended up being cooled down in the cold room. Whenever we published something in my PhD lab, we celebrated with, I want to say, at least 5 to 6 bottles of champagne. We never went
Yeah,
home before finishing those, and they were cooled off in the cold room.
yeah. Well, actually, it's a funny story. So when I went for interview in Medical University of Graz, so we had a dinner, all the students and candidates with all the faculties.
Yeah.
So and then They were serving wine and whatnot, and I was obviously drinking, and I was the only candidate who was drinking. And then Maria was looking at me. Okay, this guy is coming from Iran. He's drinking,
Yeah.
and so we clicked there. So
It's the
that's why I
drinking.
got the admission. Yeah, and yeah, so
That's good because it showed that you're relaxed, you feel confident, and you wanted to drink and have a drink.
yeah, 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 exactly.
I
Very
like that. I like that, and I think in Europe, especially in Austria, Germany, and things like that, alcohol and beer, for example, makes it's a very important part of the culture. I remember going to a GRC, and we were talking about this before we hit record. I was at the molecular pharmacology GRC in El Choco for a week, and I spent a couple of extra days in Italy. And I decided to go home to Romania to visit my family and friends. And after 10 days in Italy having pasta, my transfer was through Munich. And that morning I was like, okay, I need an omelet and I need sausage. I need something, I need meat. And I was sitting there having breakfast, and there was a bar, and there were at least 5 or 6 gentlemen at 7 a.m. drinking beer. And I was like, okay, that's one way of doing it.
yeah, yeah, yeah,
So I feel that connection as well. And being half European, I can definitely relate to that.
yeah.
All right, sorry, I interrupted you. So the other aha moment.
Oh, yeah. So again, probably I mentioned so finding that protease
Yes.
in the lung sections. I mean, of a real patient. That protease is actually an allergen by itself. It induces some immune responses. So it's not. So it is immune response, but also regulating GPCR signaling through integrins.
Integrins, yeah.
Yeah, and extracellular matrix. So yeah, that was definitely a highlight. Yeah.
You had a hunch. And you followed it. Took
And
2 years, but you followed it.
yeah, yeah, yeah.
Anything else?
And probably, finding a way to activate GPR64, the adhesion GPCR, and seeing a signal in cells that were just removed 2 hours ago from a patient. It just makes you feel, useful. Let's
Yeah.
say that, yeah, you are doing something that really matters, and it's not just pure basic science.
Yeah.
There's a meaning for it, what
Yeah.
you found for activating the receptor. So
You
yeah,
make
those yeah.
And I was going to say you make a great point, and I wanted to ask what kind of assay, what's the activation mechanism that you were able to measure?
So GPR64 was a GS couple, so we were looking at cyclic AMP by HTRF. We couldn't do kinetic because it was hard to actually transduce it.
Yeah.
Yeah.
But still, I think it's awesome, and
Yeah,
and as you mentioned, it gives meaning to the work that you do. And I've had that once with this project with Helen, whom I think I should invite on the podcast at this point because I've talked to her about her so many times. And so she's a clinician as well. And the whole idea was, can we determine the molecular mechanisms of an immunodeficiency that is due to a mutation in G alpha I2 in patients? And so we went from one patient to now many more. But basically, the bread experiments I ran and the cyclic AMP measurements I've done in HEC cells look exactly the same like the assay they've done in fibroblasts and immune cells from the patients.
okay, that's great.
And
That's great to see that they match.
yes,
Most time they don't.
they don't exactly.
Yeah, no,
And
that's fantastic.
when you read reviewers' comments, typically is it's HEC cells,
Another
it's
hexel. Yeah.
not physiologically relevant. And this was the time when I looked at the data and said, yes, finally, something that really matches, and it actually helps us map out what happens in patient cells.
Yeah. No, that's amazing. Yeah, that's one of the things. I mean, I'm not a critic of hex cells. I have used it plenty of time, obviously, but sometimes we get too comfortable with just hex cells, and we don't want to do, I don't know, iPSC or primary cells and whatnot. Um, I hope we all move toward that, toward something more relevant, to the patient.
Yeah, I think the field slowly but surely is moving towards that. I think we all acknowledge how useful hex cells are, as you just mentioned, but there are many labs that are developing more, I want to say, physiologically relevant cells, as you mentioned, iPSCs or even cardiac cells, or
Mm-hmm.
or even tools to detect endogenous GPCR expression, endogenous GPCR function.
Yeah,
And I think that is something that not only it's needed, but it's something we're going towards slowly as a field.
yeah, yeah, yeah,
And that's
yeah,
the
yeah.
beauty of it.
So, yeah, exactly. I mean, there are knockouts of G proteins, there are knockouts of beta-arrestin, there are knockouts of GRKs, but majority of them are in hex cells still.
Yeah.
And to define their function, probably we need to go to some more, more relevant systems. But it takes time. Obviously, it requires more efforts, more funding. It's much slower. I realize that. Um, but yeah, it's good to hear that there is that intention and motivation to do that.
Yeah, yeah. And also marrying, I think, a lot of animal models, not a lot of rodent models. By the way, I wanted to mention when you were talking about, nasally challenging the mice. I mean, I've worked for a very limited time with mice at NIH. It was very difficult for me. It's not something I can do, but kudos to you because your life revolves around the mouse colonies and making sure that you have the right number of mice to do the experiments, and then doing the manipulation is also challenging.
Yeah. Yeah, I mean, it was challenging, and I didn't have any experience before that. Maybe a little bit during my master's, but again, environment helps you. I had the best colleagues who would teach me things that I never thought I would be doing. So, yeah.
That's amazing. All right. Any final thoughts before I let you go?
Um, Not really, except that um, again, I would say to the trainees and postdocs, to even early career scientists, that explore. Don't think that what you are doing is something that you are supposed to be doing. There are other options. Sometime your mentor or your manager um, is a great person who can actually find your main talents even better than you.
Yeah.
And they may tell you that, hey, you may be doing the other thing much better than what you are doing, and you may actually enjoy it. So explore those things. Don't get stuck in whatever you are doing. People were telling me a few years ago that, oh, after a few years and postdocs in academia, there's no way to go to pharma. Now, when I was coming to pharma, they were saying, "Oh, when you go to pharma, there is no way to go back to academia. Don't follow
Yeah.
those advice. Just
Yeah.
just go for what you enjoy doing, and things will get together to help you."
Yeah. That's amazing advice. And I think it's about, following your gut, following your path. And so what if you're the first person who, goes from academia to industry, back to academia, back to industry? Who cares? Yeah, it's your
It's
it's
your
your
life.
life. It's your life, it's your path.
Exactly.
And you made also a great point about, listening to, for example, sometimes your mentor who can or others who see you can identify the things that you're really good at. And this reminds me of me because I was in academia for 20 years where for a long time I was focused on the things that I'm not able to do well.
Mm.
And that did not make me happy, and it felt like I was really forcing things. And that's not a nice state to be in. Now, doing what we're doing here, this comes naturally to me.
Yeah.
I love talking to people. I love this podcast. I love this. And I've shifted to majoritarily do the things that I'm really good at. And the things that I'd like to learn, keep on trying to learn. But if it's something that I don't particularly like to do, I don't want to learn, why not find collaborators?
Yeah.
People
Yeah.
who
Yeah.
are good at that, because when you think about the work that you do in the lab, you can't do it all. You have to collaborate. You have to
Oh,
talk.
absolutely. Absolutely. And that's actually much more important when you come to pharma because, I had to do everything on my own. Even if you have collaborators in academia, still,
It's not
you
the same
probably are
thing.
separated. Yeah. So when in pharma, I work with chemists, computational biologists, and DMPK, and all different uh, ways of biology, and I learn a lot every day. So don't stop learning.
Yeah,
That's what I would say.
that's
Um,
true. That's true.
but I have to say, so from the first, I'm definitely sure that I listened to the first few episodes
Hmm.
of yours in 2020, right?
Yeah.
So you
Yeah.
started sometime in April, May, something like that.
Yes, May.
Um, had you done any interviews before like that?
No.
No. Wow. So I mean, for me, I didn't know you. For me, it was like, okay, this person has been doing this for at least 10 years. No, seriously. So
Thank you.
no, seriously, what you just said, that you feel comfortable doing this and you know that this is exactly what you want to do. This is what I was talking about. So explore different things.
Yeah,
Don't
thank you.
don't get stuck. Yeah.
I never did this before, and I think I talked about this with other guests as well. So the idea of doing the podcast came out of a need for me to contribute to the GPCR field without pipetting. I
Mhm.
worked with hex cells, and my life was dependent on how my hex cells looked like, whether I could run the plates or not run the plates, or whether
Yeah.
my trainee or my RA, split the cells correctly, and it would just drive me nuts because he didn't listen to me or she didn't listen to me and things like that. And the first, I want to say, first two episodes, I don't remember after the first two, but the first two were nerve wracking.
Oh,
I
I
was,
I I did not notice.
thank you,
No, I don't think anybody noticed. It
thank you.
was quite, no, seriously.
The first one was with Paul Incel, and Paul, oh my God, he talked so much, and I'm so glad because everything he said was super interesting. But in my mind, I was like, Paul, keep on talking.
Keep talking.
I'm super nervous. And then the second one was with Terry, and Terry talked less than Paul. And inside my inside voice was like, okay, Terry, you have to expand on that. But since then, I always come into these calls energized, and I leave them 10 times more energized because
Yeah,
I get to know someone.
yeah.
And, I've made friends by talking to people on the podcast. I've got introductions. I mean, I've heard about you from Maria when I recorded my episode in the closet with her. And she said, you
Yes.
have to talk to Nariman. You have to talk to him. And here we are. Okay, it took us about 2 years for me to get to that point, and then inviting you and adjusting schedules. But I've met amazing people that I would have never met had I not started the podcast. If I had stayed pipetting in the lab on my own, then we wouldn't be here.
Yeah, and there's nothing wrong with pipetting. I
No.
want to I want to be sure everybody hears this.
Yes.
Don't think that their work is not valued. It's just maybe you can do other things much better. Just remember that. That's the
Yeah.
whole point.
I think you make a great point about, there is nothing wrong about pipetting. There is something wrong about pipetting that when it makes you unhappy.
Yeah, yeah. Yeah, be
When
happy. Be happy when you are doing something, whatever job you have.
exactly.
Yeah. Yeah.
Exactly. And I loved my time in the lab. I was ecstatic when, you run 10 breath plates and then you sit down and then you quick copy and paste into Prism and the curve looks great, or it's 9 p.m. and
Yeah.
you're developing your film and you have finally a band on it.
Yeah.
But I got to a point where I was like, this doesn't make me happy anymore. This is more stressful than it should be.
Yeah.
And
Yeah.
what we're doing here is the best thing. I could do this every day, all day long.
Awesome.
All right, we're
That's
gonna
great.
stop here. Thank you so much for your time today, and I can't wait for people to hear this episode. I think they're gonna learn a lot of fun things.
Thank you very much for having me.
Thank you.
Bye.
Bye. Thank you for joining us and listening to this Dr. GPCR podcast episode. I'd like to thank our team members, Attila Forres, Ines Pinero, and our newest Dr. GPCR proteges, Montserrat Avila Zozoya, and Nipuna Wirusingo. Welcome to the team, Monsi and Nipuna. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Email us with any questions or suggestions at hello@drgpcr.com, and please become a site member today at drgpcr.com. And until next time, stay safe.